Welcome. My name is Loni Schumacher, a veterinarian finishing my master's with the K-State Swine Nutrition team. Today, I will be talking about my two Midwest meeting presentations. The first of which is utilizing feed sequencing to decrease the risk of porcine epidemic diarrhea virus cross-contamination during feed manufacturing. In our previous research, we established a minimum infectious dose. The key finding is that when virus is mixed into untreated feed, it had a detectable Ct value, and that resulted in infectivity. We have since replicated this model in numerous experiments. An extremely low dose of 112 virus particles was found to be infectious. This translates to 1 gram of infected pig feces that can potentially contaminate 20 semi-loads of swine feed. During the minimum infectious dose study, negative feed was sequenced after each dose was mixed and evaluated by PCR assay. We found out that cross-contamination occurred. A PCR Ct, or cycle threshold, value of 38 was obtained in feed that was mixed after the most concentrated batch of PED was contaminated swine feed was produced. This, however, Ct value was much higher than detected in the previous batch of feed. Therefore, our hypothesis is that strategically sequenced diets may reduce the risk of PED cross-contamination. Therefore, our objective was to determine the efficacy of sequencing to minimize the risk of cross-contamination in a feed mixer. We utilized the K-State Cargill Feed Safety Research Center, or FSRC, which is a 3-story BSL-2 biocontainment lab that contains mixing, pilot mixing, conveying, and pelleting equipment. This study was done in 3 repetitions consisting of feed inoculation, feed manufacturing, feed mill decontamination, testing and evaluation of baseline that was done before each replication. We used a PED prototype strain that has been proven in several other experiments to be pathogenic and infective to baby pigs. Therefore, we utilized the stock virus and mixed it with 4.5 kilogram batch of feed, and that was used as our inoculum material in the positive batch. And we had a goal of a target Ct of 30. After the negative was mixed and discharged to charge the system, and after the positive batch was mixed and discharged, we then mixed and discharged a batch of feed called Sequence 1, which consisted of 50 kilograms of PED-free gestation diet. This was followed by another batch called Sequence 2, and so on and so forth, that up to 4 sequences. This mimics diet sequence during a commercial feed mill operation, and none of the equipment was cleaned in between batches of feed. Therefore, in each treatment, they were mixed for 5 minutes in the mixer. You can see a picture here in the lower right-hand side. That feed was sampled, and then the mixture or the treatment was discharged into the bucket elevator for 10 minutes in sequential order. And also, just to remind you, there's no cleaning between batches. Areas that we sampled include from the mixer and also from the bucket elevator. After the sampling was completed, we took 100 grams of each feed treatment, and that was mixed with 400 milliliters of PBS and was refrigerated overnight. The supernatant was formed and collected by following Iowa State University's bioassay protocol. We took 10 mils of inoculum and was gavaged into 3 pigs per treatment. So with 3 replicates, we had 9 pigs total, and then also a non-inoculated control. Here is the results of the data that we obtained. Along the vertical axis, we have the response criteria, or percent of feed with detectable PED RNA. And along the horizontal axis, we have our feed treatments, starting with after negative, after positive, and then sequence 1 through sequence 4. As you can see from this data, our negatives remain negative, PCR negative, and after the positive batch was formed, both the mixer feed in the orange bar and the bucket elevator feed represented in the purple bar, 100% of feed samples had detectable PED RNA. There was a decrease in the mixer feed after sequence 1, but the bucket elevator feed remained 100% of detectable feed. After sequence 2, none of the mixer feed had detectable RNA. However, there was a significant decrease in the bucket elevator feed. And after sequence 3 and 4, there were no more RNA could be detected from these last 2 feed treatments. Here are the CT values from the samples that had detectable RNA. As you can see, the mean CT values between the mixer and the bucket elevator feed are similar after the positive batch and after sequence 2. No RNA was detected from the mixer samples after sequence 2 through sequence 4. And as you can see, the bucket elevator CT value rose higher in sequence 2. Therefore, sequencing increased the CT values from the mixer and bucket elevator until no RNA could be detected from these last 2 feed treatments. Here are the bioassay results from the feed that was taken from the mixer only in this case. Along the first column contains the feed treatments from the mixer, and this included negative, positive, sequence 1, and sequence 2. Sequence 3, and we will be expanding our bioassay to include mixer samples from sequence 3 and 4 and from the bucket elevator later on. And that's something I will explain in my next presentation. Note that each row represents a room that contains 3 pigs per treatment. That contains a room of 3 pigs from each rep. The values are reported as CT values, where 45 is beyond the detection limits of the assay, and low numbers indicate greater amounts of virus was detected from the PCR assay. Fecal swabs were collected from day 0 to termination of the study, as well as cecum contents at necropsy. From this data, you can see that our negative samples remain negative, and in our pig fecal swabs were negative. With the positive, you can see that from day 2 to termination of study, the pigs broke with PED and had fairly low CT values as compared to feed. In the sequence 1 group, you can see that we had 1 room of pigs that had a inoculum with a fairly high CT value. 1 out of 3 pigs broke with PED at 2 days post-inoculation, and then by termination of study, all pigs in that room were infected. However, one of the most concerning aspects of this data is from this room, where this group of pigs received PED negative inoculum, or had inoculum that did not have any detectable RNA. However, 1 of the pigs broke with PED, and at termination of study, all of the pigs eventually had positive CT values. So it appears that inoculum can be infectious beyond the detection limits of PCR, and this has been reported in other studies, such as PED tissue homogenates at University of Minnesota and tissue culture using PED at Iowa State University. Additionally, PED infection was further confirmed by histopathologic lesions typical of PED, as well as positive immunohistochemistry. In this photomicrograph, we can see immunoreactivity in enterocytes highlighted in brown, which indicates viral replication is taking place. Additionally, intestinal morphology was evaluated. In this bar graph, the vertical axis is the villus height measured in micrometers, and along the horizontal axis, we have our PED feed sample challenged pigs. And as you can see, there's a significant difference and decrease in villus height from the positive pigs, as you would expect in PED-infected pigs. Additionally, when we looked at the villus height to crypt depth ratio, we had a decrease villus height to crypt depth ratio in our positive pigs as compared to our other feed treatments. Therefore, conclusions from this study are as follows. The sequencing protocol can reduce PED carryover. Feed without detectable PED RNA can be infective. We plan to expand our bioassay in 2016, funded by the National Pork Board, where we're going to look at the mixer samples of sequence 3 and 4 and place those in pigs, challenge pigs, as well as challenging pigs from the bucket elevator samples. Additionally, it appears that contaminated feed mills are a potential route of PED infection into farms, something to consider when formulating a biosecurity plan. Finally, I would like to acknowledge our partners in PED research, which include the K-State Grain Science, K-State Applied Science Nutrition Team, K-State Veterinary Diagnostic Lab, and the Iowa State University Veterinary Diagnostic Lab. Additionally, appreciation is expressed to the National Pork Board for financial support. Please stay tuned for my next presentation, which is the Midwest abstract presentation entitled Evaluating the Effect of Manufacturing PED Contaminated Feed on Subsequent Feed Mill Surface Contamination. Thank you.